Good morning, sister chromatids. This is MRP4, your daily reproduction broadcast. Today, I, Lizzie, will be showing you the wondrous world of meiosis, specifically meiosis 1. Starting off with prophase 1, the nuclear envelope in the diploid germ cell disappears and the chromosomes begin to condense and form. Synapsis then occurs when the homologous chromosomes loosely pair up, aligned by the type of gene. In crossing over, the sister chromatids exchange DNA segments like such. Chiasmata, an X-shaped region, is also formed where the crossing over occurred. The centrioles then begin to form along with the spindle fibers and start to move toward the poles of the cell. Next, metaphase 1 begins, and the tetrads line up at the metaphase plate. The homologous chromosomes orient randomly at the plate, known as independent assortment. This contributes to the genetic variation in sexually reproducing organisms. In metaphase 1, the spindle fibers also attach to the centromeres. Next, in anaphase 1, the pair of homologous chromosomes separate from each other and move toward each pole guided by the spindle fibers. As this is done, the cleavage furrow begins to form. In telophase 1, each half of the cell contains half the set of chromosomes. Cytokinesis simultaneously occurs, forming two daughter cells. And that's it for meiosis 1. Wasn't that quite the adventure? But don't fret, the roller coaster is not over yet. Take it away, Sister Surekha.
Thanks, Sister Lizzie. What a darling she is. Now, folks, on to the second phase of meiosis. We start with prophase 2. Within prophase 2, the two daughter cells produced from meiosis 1 have their nuclear membrane slowly disappear, their chromosomes decondense, and the cell has new spindle fibers form around the chromosomes. In metaphase 2, the spindle fibers are fully formed and each sister chromatid ends up forming an individual kinetochore that attaches to microtubules from opposite poles. The centromere functions to provide a foundation of assembly for the kinetochore. Basically, the sister chromatids are condensed and aligned at the center of the cell. In anaphase 2, the sister chromatids are pulled apart by the kinetochore microtubules and move towards opposite poles. The non-kinetochore microtubules are there to elongate the cell. In telophase and cytokinesis 2, chromosomes arrive at the poles of the cell and end up decondensing. The nuclear membrane forms again surrounding the four nuclei. The cleavage furrow divides the originally two diploid cells into four haploid cells, which can also be considered four gamete cells. Thank you so much for tuning in to MRP4 News. We will see you next week where we will be diving into the coronavirus.